Riccia introduction. It is most widely distributed genus of family Ricciaceae belonging to class Hepatocopsida of division Bryophyta, having almost 200 species. About 30 species are found in Pakistan. The most common and worldwide distributed species is Riccia frosti. Habitat. The species of Riccia prefer to grow on damp soil, moist and shady rocks and other similar habitats. However, a single species is reported as free-floating on stagnant water or slowly running water. Structure. External structure. The plant body is dichotomously branched, dorsiventral and prostrate thallus. The branches of thallus are called thallus lobes. Each lobe is thickest in the middle and gradually thins towards the margins. The thick middle portion constitute midrib region. The thallus is attached to substratum by slender, simple, unicellular structures called rhizoids. These may be smooth-walled or tuberculate. Their function is anchoring and absorption. In submerged species, rhizoids are generally absent. In addition to rhizoids, multicellular scales are also present on the lower side of thallus. They are situated closely to the growing point and protect it. Internal structure. The thallus of Riccia shows an internal differentiation of tissues. It shows two distinct regions, a ventral region or storage region. This region is formed of a compact colorless parenchymatous tissue without intercellular spaces and often contains starch but no chloroplasts. The lowermost cells of this region are small in size and regularly arranged to form lower epidermis. Scales and rhizoids are arising from it. This region serves as the storage region of thallus. B. Dorsal region or assimilatory region. This region is composed of green, chlorophyllous cells that are separated by vertical air canals. The canals are open on the dorsal surface so that the top of the thallus is porose. The outermost cell is larger and colorless, which forms one cell thick epidermis called upper epidermis. This region, also known as photosynthetic region or assimilatory region of the thallus. Life cycle or reproduction. Vegetative reproduction. It takes place by following methods. Fragmentation. The cells in the older portion die and eventually disorganize. The young lobes become separated and grow into a new thallus by apical growth. Adventitious branches. These branches arise from the ventral surface of the thallus. They become detached from the parent thallus by the decay of connecting tissues and form new thallus. Tuber formation. The apices of thallus become thickened to form tubers at the end of growing season. These tubers remain dormant under unfavorable conditions and resume growth under suitable conditions. Sexual reproduction. It is always of oogamous type. Species. Homothoric or heterothoric sex organs. Antheridium, male, Archegonium, female, alternation of generation, regular and heteromorphic gametophytic phase. So it is the dominant plant phase, represents haploid generation and develops sex organs. The gametophytic phase begins as haploid spores are formed. Sperms and egg are the last structures developed during this phase. Sex organs are developed on the thallus lobes. In the monoecious or homothoric species, the antheridia are formed first and archegonia later. Gametogenesis. Antheridium. The mature antheridium is an elongated structure. It has a narrow pore or opening at upper surface called osteole. The body of antheridium has an outer jacket layer of sterile cells called antheridial wall. This wall is protective in function. The antheridial wall encloses a mass of small fertile cubical cells called androcyte mother cells. Each androcyte mother cell has a denser cytoplasm and a larger nucleus. It divides to form two sperm cells known as androcytes. The protoplast of each androcyte metamorphosed into a single sperm. Each sperm is a minute, slender and curved structure having a pair of whiplash flagella at its anterior end. The sperms do not release from antheridium until enough moisture is present to permit them to swim over it. Archegonium. It is flask-shaped structure and consists of two parts. Venter. The basal swollen portion is called venter. It is directly attached to the tissues of thallus. It has a jacket of sterile cells called venter wall. It encloses the venter cavity, which is filled with two cells, upper and smaller ventral canal cell, lower and larger egg cell. B. Neck. The long and slender portion is called neck. It consists of four neck canal cells surrounded by a layer of sterile cells called neck wall. When the archegonium reaches maturity, the neck canal cells and the ventral canal cells degenerate to form mucilage. The mucilage imbibes water and swells. A neck canal is formed through which sperm enter into venter that containing egg. Fertilization. It takes place in the presence of water. When a sufficient amount of water or moisture is present, antheridium bursts and releases the sperms. Usually, the heavy dew provides sufficient water to permit movements of the sperms. The mucilage oozes out from archegonia and attract male sperms gemotatically. Many sperms move towards egg, but only one of them gain entrance into the venter through neck canal and fuse with egg to form zygote. The act of fertilization ends the gametophytic generation and is the beginning of sporophytic generation. Sporophytic phase. The sporophyte of Riccia is simple and completely dependent on gametophyte. It lacks foot and seta. It consists only of capsule. The sporophytic phase represents diploid phase and begins as zygote is formed after the fusing of egg and sperm. The zygote increases in size and undergoes repeated cell divisions to form embryo. It almost completely fills up the venter of archegonium. The walls of the venter divide pericolonally and form a two-layered calyptra around the young sporophyte. Sporogenesis. The superficial cell of sporophyte capsule divides to form an outer amphithecium layer and inner endothecium layer. The amphithecium forms the jacket of sporophyte while the endothecium represents the archosporium. The archosporial cell divides to form spore mother cell, sporocytes, and sterile nurse cell. The sporocytes are the last cells of sporophytic phase. Nurse cells and amphithecial jacket layer disintegrate. Sporocytes undergo meiosis to form haploid spores. Spore is the first cell of gametophyte.